Ok, è partito il live su Zoom, su YouTube.
no. No. Apparently now Silvio Tosatto is the host. Because after it crashed randomly, he has been assigned again.
Yes, I don't know. It happens sometimes.
You want me to assign someone else as host?
Yeah, you can assign again. Let's try that. Because, okay. When I started the live streaming service, okay, now I'm the host. Okay, great, thanks. I think we can start.
Wonderful.
Okay. Good morning. Claudia, are you there?
Again.
Buongiorno a tutti.
No.
Okay, welcome. Thanks and sorry for the delay, but we had some technical problems. Hopefully now everything is working. Okay, so according to the guidelines that you got, you have approximately 20 minutes. So let's say 25 minutes, including the question and answer. So now you can start. You can share your screen, and hopefully now it's working. Can you do it?
No.
No. Okay, wait. Okay, let's try now.
Okay.
Great, perfect. Okay, now you can start.
Okay, perfect.
Thanks.
This project focuses on automated learning of planning domain. Automated learning is a branch of artificial intelligence that aims to support the learning activity by reasoning on conceptual models that are abstract and form a representation of the world, called planning domain. In this abstract representation of the world, there are agents that perceive the real world through sensor. And it is part of the ability of the agent to fill the gap between these two levels. So the abstract world defined by the planning domain and the real world perceived through sensor that return typically data on real number. The aims of this thesis is to use automated planning to build abstract planning domain by observing the effect of action on the environment. This master project takes place within the research activity carried on by the Fondazione Bruno Kessler in Trento in Data and Knowledge Management Unit. The work on planning and learning developed so far in this research activity captures this idea of two levels, so abstract model, described by the planning domain, and real world. And they propose an algorithm, the acting and learning planning domain algorithm, that interleave planning, acting, and learning, and they build a finite planning domain. The current version of ALP algorithm comes with some limitation. So the states are atomic, and to learn the effect of an action, we have to physically perform this action on the real environment. The new contribution of this thesis are to extend this ALP algorithm to a factored planning domain, so in which state are not atomic, but are represented by a set of state variables. Then after that, we to deal with the problem of learn entirely the planning domain, we extend the ALP algorithm with a prediction part. So using neural network for predicting the effect of an action in environment. Then we implement this algorithm and then we test it in some simulators. And then finally we evaluate the learned planning domain. First of all, I want to give a definition of planning domain. So planning domain is composed by a set of finite state which correspond to the possible configuration of the world. Then we have a finite set of action and a state transition function gamma. That is a function that maps a couple, so a state and an action, to a state. And this state is the result of applying this action to that state. The model assumption are that we are in a finite abstract state, so the set of state and the set of action are finite, and then we have the determinism. So any action taken in a state has one successor state, so the mapping is to one single state. Now we want to see the first contribution of this thesis is to extend the ALP algorithm, so the set of state, to state that are represented with state variables, so a vector of value taking values over some domain. So let be a vector of m state variable and d a set of domain. Then we have a function that maps each state variable to a domain. And the cross product of all this domain for all the state variable is the set of the total assignment. And the set of state is a subset of this set of total assignment. The agent perceives the world through a vector of continuous variable that are called perception variable. So we have n variable ranging over real number, and we can define a function, the perception function, that maps between the state variable and the perception variable, and give us the likelihood of perceiving some perception variable given a partial assignment to the state variable. This perception function is defined as a factored conditional probability, and each function pxj belongs to a parametric family of parameter theta xj. Now we see an example of a planning domain. So we use some simulators. So in this case, we have a grid, and this grid is divided into cells. And this, in a real implementation, this grid may be flat, for example, and the cell are rooms. Inside, we have a robot, and this robot can move in this room. In this case, we have some closed room. So, for example, in here we have a door, and in here we can have an elevator or a wall. So, in this case, the planning domain is the set of state are the rooms, and then we have the action. So the action are north, south, east, and west, and will cause the robot to move one unit in the action direction, so to move in the adjacent room. In case that the action we cannot perform this action because of the wall or a door, then the robot remains stationary. The robot perceives the world through sensor. So in this case, the sensor return a vector of 6 variables that are the x and the y coordinate, and then we have the distance between the wall in the 4 direction. There may be situation in which the abstract world is not sufficient. So, for example, we can take the example that the closed room are open. So now the robot may perform this action and may end up in a different part of building. So, in this case, the set of state, if we have a fixed set of state, this may be not inadequate to describe this new situation. Instead, if we have a dynamic set of states, so if we can add some state during the iteration of the robot, then we can deal with an expected situation and a new state. So we will use a dynamic set of state, and we will see an algorithm that learn the transition, so the state transition function, learn the state, so extend the domain of the variable, and then also update the perception function and update the transition function. So now we see in detail this algorithm, so the active learning planning domain algorithm. As input, we have an initial planning domain, so we start from an empty planning domain. Then we have an initial perception function and an initializer. This is a method to initialize the parameter theta for the perception function for every perception variable. Then we have max iter, that is the maximum number of iteration, and then we have these three parameters, alpha, beta, and epsilon, that allow the agent to define its behavior. So they represent the key component of the agent, of the algorithm, and they tell us how much the agent trusts in the abstract model. So alpha for the transition, beta for the perception function, and epsilon for the state. So first of all, we have an initialization. So we have this two set. This set is for the history of the observation, so the observation, and then we have the transition. And first of all, we have this sense, this function return the perception variable. So through the sensor of the robot, for example, we have a vector on a real number. Then we call in this function best current state that in this first initial step, it only define the first initial state, because we start with an empty set of states. So first of all, we have the initial state. Then we go into the Y cycle. So we select an action. So between the action A, we select an action. So we can select an action randomly or using a plan strategy or an explore strategy. Then we execute the action. So we execute physically the action. So the robot performs this action A in the real environment. Then we sense that means that through the sensor we return the perception, the vector of the perception variable, the value of the perception variable. Then we have the best current state. So using this function, we go to search in the set of state if there are a good state. So using this perception variable and the perception function, we calculate the perception function for each state and we select the best one. If there is no a good state, so there is no state above some threshold, then we have to extend the domain. So which domain? We calculate the perception function, and the variable that are under the threshold, we have the domain of this variable, and we calculate the minimality set between this variable. Then we bring up to date the set of observation, the set of the transition, and then finally we have to update the state transition function using the old transition function and the new transition, and then also we update the perception function. To update the perception function, we go to update the parameters theta of the perception function. So we will use the old parameters and the new observation. So now we have seen this algorithm that builds a planning domain. But now we want to see some computational aspect, because we have seen that to learn the state transition function, we have to physically perform this action. So the robot have to perform the action, then we sense, so to the sensor we have the perception variable, and using the perception variable we analyze the state and we select the good state, or we extend the set of state. But so the ALP algorithm represent all the state transition function with explicitly, so with a matrix or a graph and so on. But if we are in the case of a huge amount of state and action, we are not able to perform all the action in all the state to complete entirely the state transition function. So the idea is to represent gamma as a neural network to use the prediction, so predict the effect of action on the environment and use this prediction to complete the state transition function. So the idea is to use the structure of artificial neural network. So we train a neural network for each action using the data. So we have the perception variable before executing the action and the perception variable after the execution of the action. Using this data, we can train a neural network for each action. And when we have the difference between the state predicted, the perception variable predicted by the artificial neural network, and the real perception variable perceived through the sensor, the difference is under some threshold, then we can use this neural network to go to complete the state transition function using again the perception variable and so on. So some result in the simulators. So we have to compare the prediction of the neural network with the real perception variable, so perceived through the sensor. So in this case, we have the robot. So the simulator is the robot with two perception variables, so the x and the y coordinate. Instead, in here we have again the robot, but we have the 6 perception variables. So we have 6 sensors: the coordinate x and y, and the distance from the wall, and we can see the loss function, the loss that it decreases. So the neural network have learned in this case the function as list, the action list. So the last thesis contribution is to go to evaluate the learned planning domain. So we want to see if how good is the learned planning domain. So we have to measure the state variable state space and the state transition function. So here I report a measure for the state transition function. So we have the coherence. So a good model is when we have a coherence between the abstract model and the real world effect. And since we have that the state transition function is a deterministic function, so when we have a couple, so a state and an action, then it maps into one single state. So if we have some discrepancy between the state predicted by the transition function, for example, if we are in state S1, we perform the action A, the state stored in the state transition function say that we go into the state 2. But if we the robot perform this action, and through the sensor and the perception variable, the state that maximize the perception function, so the state that come out from the analysis of the perception, the sensor variable, is different from this state, then we have an incoherence between the abstract world and the real world. So we want to measure this incoherence and discoherence. So in here we have an example. So again, in the robot with 6 sensors. So we can see that the coherence in this case is 0.89. Thanks.
Okay, thank you. So are there any questions from the committee? Okay, I have one in the meantime. So when you introduced more or less around slide 12, 13, so when you talked about the effect of the perception function on the, performances, and then how you compare the abstract, let's say, the theoretical performances with what you get from the real perceptions outcomes. Okay, my question is, the very general one is if you can add a few words about the impact of this of the output of a real perception function that in most cases, I guess, is it's very noisy. So if in your experiment, did you see huge differences from that point of view? And how much do you think that your model is robust with respect to a realistic scenario where you get, I guess, something that is mostly run on top of what you get from, for example, vision-based sensor that is very noisy.
Yes, we have some noise. We add some noise in the simulator for the sensor, and this we to deal with this perception function, we try to make the model robust, so that is able to deal with some noise. So when, for example, we have some noise in the perception variable, and when we add we update the parameter theta for the perception function, we take into account this problem of noise and sensor error. And also in the experimental evaluation, we try to test this topic, because when we go to an experimental evaluation in the part for the evaluation of the state space, so the set of state, then we also see that we compare the state that we have learned using the perception function, and we see that in some cases there are some redundant state, so some double state that is called causes for this noise, this error. And but if we can give a good threshold for the when we choose the state. So if we are we find a good method to define the threshold to have a good result to contrast this noise.
Okay, thank you. Are there any other questions? No. Okay. Time for the last question? No. Okay. So we can move on. Thanks. Thanks a lot. So we'll see you later on after the next candidate.
Okay.
Thanks. So let me admit then Árpád.
Posso rimanere qui o esco? I'm sorry. Can I stay
Up to
here?
you. I mean,
Okay.
okay. Árpád, are you there? Hello?
Good morning, everyone.
Okay, good morning.
Good morning. Do you see and hear me?
Yeah, now yeah.
Yes, awesome.
So welcome. You should be allowed to share your screen. So please do it.
Yes, I will try to. So, right, share screen. Hopefully. Yes. Do you see the presentation?
Yeah.
Yes, great.
Okay. So you
Thank you.
start. You have 20 minutes. Okay.
Thank you. So, welcome everyone. My topic of research was prediction of health status, specifically that of inflammatory bowel diseases, based on the composition of the human gut flora or gut microbiome. So the motivation for this is that there is an emerging industry of personalized preventive health consulting or personalized medicine, which requires a data-oriented approach for predicting clients' and patients' health status and disease risks based on various kinds of data obtained from medical examinations. One such source of data is the human gut microbiome, which is just basically the composition, the different species of microbiota that live in the human gastrointestinal system. And it's very rich in data. There's a lot of species, a lot of kinds of lots of genes, but it's still not very well characterized. IBDs, inflammatory bowel diseases, and the related family of diseases called IBS are quite serious diseases of the gut and the intestines. So it's only natural that we want to predict health status or the presence or the risk of developing IBD or IBS from the composition of the gut flora. So here I would like to explain what metagenomic analysis is. It's basically the analysis of all the genetic material, like DNA or RNA, that is given by the composition of the microbiome. So we take samples from the human gut, for example, fecal samples, and then we sequence them, and then using the RNA or DNA obtained, we will perform our analysis. So far, most of the analysis or metagenomic analysis of the human gut have focused on quantifying taxa, so species or strains or genera. But the problem is that biological functions that lead to diseases or lack thereof are associated more with genes rather than taxa. So our approach, the novelty on our approach is that we started using genes corresponding to certain functional groups rather than taxa for the analysis. So here you can see a brief overview of the workflow. First, we collect fecal samples from clients to be examined. Then these fecal samples are used for sequencing the DNA content. And thus we obtain millions of short genomic reads. And then we quantify these reads using some method or another. And for each client and for each taxon or gene, we obtain a so-called relative abundance, which is just a fancy word for the frequency of a given gene. And then we use this data for training a machine learning model, so supervised learning. We use this for training a machine learning model, and we aim to classify healthy versus IBD clients by means of generating an IBD score between 0 and 1, where 0 means healthy and 1 means IBD. And then, of course, since we have performed this work in the context of a personalized medicine company, we will use this data or this score to visualize and explain the data to the clients. So for this purpose, we have developed a metagenomic analysis pipeline. Our biologists have composed, based on the literature, 121 groups of homologous gene families. And these contain genes that correspond to very basic and fundamental microbial functions, biological functions. The first step of the software that we have developed basically takes some configuration and downloads these gene families, and then performs the multiple sequence alignment on them in order to build HMM profiles from the gene families. And these HMM profiles will later be used for searching for the reads obtained from fecal samples. So for quality control reasons, we are outputting some visualizations after building the database so that experts like biologists and bioinformaticians can examine the contents of the database. And for example, in this picture, we can see that this is a very good quality sequence alignment of a highly homologous, not very distant family of genes. And this means that the resulting database makes sense. The second step of this software that we have built is actually concerned with quantifying the relative abundances of the genes that appear in the genetic content of all the microbiota of the human gut. This is achieved by aligning reads to either the HMM profiles that have been built or directly to the reference genomes or genes that have been downloaded. Either of these methods can be used, or both. And then we count all the reads, all the DNA reads that mapped to one of these references, and then these could be normalized then in order to obtain relative abundances of certain genes. So this is basically just an example. This is a big table where a couple of clients have their genes quantified. So rows are clients and columns are genes, functional gene families, rather. In order to analyze relative abundances, we have to consider a pretty serious statistical problem with it, the fact that it is compositional, which means that it sums to a constant, and without a loss of generality, we can assume that they sum to 1. This means that there is a missing degree of freedom in the data, which means that naive classical statistical analysis of the data, for example, computing correlations, cannot be performed because there would be a lot of spurious correlations. This means that we have to transform the data somehow, and some statisticians, for example, Acheson from the early 21st century, proposed some methods. We have 3 log transforms. These are transformations based on log transforms that we have performed. There is centered log ratio, or CLR, and relative log expression, or RLE, which are based on either column or row-wise comparison of each relative abundances to their column or row-wise centrality. And we are using the interquartile mean as a centrality measure because it's very robust to outliers. And there is another one proposed by Acheson, I think, which is ILR, the isometric log ratio, which is just the central log ratio transform of the data multiplied by an orthogonal matrix HD, which is, however, missing a row. This means that this transform basically completely removes the missing degree of freedom, and therefore the data will make more sense in order to analyze it. All right. After we have transformed our data, we have performed exploratory analysis of it, of course, as a first step of modeling. So basically, we have used 4 studies in order to obtain relative abundances, 3 big open access studies, for example, Lloyd Price's study, or studies from the American Gut Project. And we have also at our company, MediPredict, we have also 15 clients that have been involved in this study. So based on the exploratory analysis of this data, unfortunately, it doesn't appear to cluster very well based on the disease status of the clients. Here we can see TSNE, which is an unsupervised dimensionality reduction algorithm applied to one of the data sets that we have obtained. And the data points are colored based on disease status, and we can see that there is no clear clustering. However, if we color the points based on the study that they have been obtained from, then we can see very clear clustering, which unfortunately means that the biological signal is so weak that it is being suppressed by the differences between experimental methods between labs. So this suggests that it's going to be a hard problem classifying them. And of course, this means that in this picture, we can see, or in this plot, we can see correlations between the target variable, 0 or 1, and the individual genes. And we can see that the correlations are, of course, not very high. So no individual gene is able to, no individual gene's presence or absence is able to explain clearly disease status or health. So because we have a quite small data set, it takes a lot of time and number crunching to obtain data just for one client. So we had time to obtain data from about 500 clients, a little more than 500. So this means that we can't use any very complicated models such as a neural network because there is not nearly enough data. So we need to turn to simple models, and we chose to use logistic regression because it's quite easy to explain and it's quite fast to fit. And there is a clear method for regularization, which will be needed later. Because the data is high dimensional, the number of rows is comparable to the number of columns. So we will need to rely heavily on regularization. And it's nice because logistic regression is a generalized linear model, so we can use the elastic net regression or the elastic net regularization model. Furthermore, because clustering is not clear based on just the data itself, we will need to turn to feature engineering tricks. And most importantly, we need to add interaction features. So we have built machine learning models using scikit-learn, specifically the pipeline class, which consists of normalization and scaling to 0 median and unit interquartile range of the data. Then we perform some dimensionality reduction before adding interaction features. Then we add interaction features. Then this resulting data is fed into the elastic net logistic regression model. And finally, the whole pipeline is optimized using the skopt packages based search CV. So it's a cross-validated optimization of the hyperparameters. All right, so results. We have compared the results of models trained on our dataset versus models trained on a dataset that is produced by Shogun. Shogun is a state-of-the-art metagenomic analysis pipeline. But it is based on taxa, so strains and species rather than genes. And we can see that the prediction quality, as assessed by the ROC area under curve measure, is quite good. It's 0.09. Sorry, it's 0.93. And we can see that the confusion matrix is also quite impressive. The model has an overall 12% false negative and false positive rate. Compare this with our model. Well, our model performs slightly worse. However, but it's still not bad. It's still significantly better than chance. And there are several advantages. First of the first advantage is that it seems to be more robust or less prone to overfitting. Thinking about it, it's not very surprising because when we are considering all the species in the human gut microbiome, we are considering thousands of features. So a dataset output by Shogun literally has, for example, 6,000 or 8,000 dimensions. However, our dataset only has 121 dimensions. In this plot, we can see the points of the decision boundary that have been obtained from scikit-learn in order to draw the ROC curve. And now we can see that the 6 models on the left-hand side are models trained on Shogun's data, and the one model on the right-hand side is a model trained on our pipeline's data. And we can see that the decision boundaries are a lot more spread out in the case of models that have an overall lower score, which means that these models are a lot more confident in their predictions, which suggests that a higher score actually in this case means that the other models, at least the three on the left-hand side, are overfitting. So our model is less prone to overfitting. Just while we are under an overfitting, it's nice to explain feature importance. The problem with adding interaction features and normalization and regularization and so forth is that the clear connection between the composition of the microbiome in terms of relative abundances of genes is lost because they are not the direct output of the logistic regression model. However, we can use something called permutation importance in order to still evaluate the importance of each feature. This means that each feature is being shuffled randomly, one by one in the dataset, and it is evaluated how much predictive power the model loses when this feature is being randomized. And of course, this plot doesn't say much in itself, but if we turn to experts in biology and show them which genes turn out to be significant or highly important, then we can draw further biological conclusions from this. Another advantage of our software compared to Shogun is that, of course, if we are relying on only a couple hundred genes, which are short sequences of a couple hundred or a couple thousand base pairs, then we will need significantly less storage space in order to store the database, the reference database. Shogun, of course, uses a huge reference database with thousands of species and strains composed of whole genomes. So its database takes about 100 gigabytes. Compared to this, our database takes about 2 gigabytes or 2.5 gigabytes. Furthermore, we are using in our pipeline, we compose this software of state-of-the-art read alignment software, for example, Minimap2, which is very, very fast, which resulted in the additional advantage that our analysis is a lot faster to run too, compared to Shogun. There is still a lot of room for improvement. For example, we needed to take some shortcuts during this work in the interest of time. So this means that the methodology needs some overall improvement. For example, while aligning to HMM profiles, while aligning metagenomic reads to HMM profiles, it is necessary to subsample these reads because HMM profile alignment itself is significantly slower than raw read alignment. But subsampling, of course, introduces the problem that very low abundance species or genes will be completely missed from the resulting profile. So we will have heavily zero-inflated data and heavily skewed data. And this needs to be overcome either by means of some additional statistics or by means of not relying on subsampling and instead obtaining enough computational power in order to complete read alignment to HMM profiles. There are also quite some naive algorithms in the post-processing of the data, of the read counts, which are quite slow as they are implemented in Python. They could be either re-implemented in C or something like that, or improved algorithmically, or both. It is also very interesting to see why the models are making mistakes and what mistakes they make. And for example, it's like just a curiosity why some models overfit so badly and why others perform better in terms of robustness. So for this, we could turn to visualization or explanation libraries like SHAP or ELI5, which are Python libraries for explaining the output and the behavior of the scikit-learn machine learning models. So basically, that's what I would like to say today. And if there are any questions, please feel free to ask. Thank you very much.
Thank you. Thanks for the presentation. So we have time for questions.
I think I can start probably with a question. So first of all, very interesting work on, and also very timely work on the relationship between the gut flora and various diseases, in this case, the irritable bowel ones. So my question would be, since it is well known that the gut flora influences the irritable bowel disease and syndrome, but so depending on what you have, obviously, this can get worse or it can get better. On the other hand, there are also genetic components to these types of diseases. So my question would be, have you taken into account the also the genetic components of your patients? And if not, can you estimate how much that would influence your results?
Thanks for your question. So basically, we are not currently taking into account the genetic components in the read alignment or the abundance counting part. However, actually, at the company that I did my work for my thesis, we do have a separate genetics team and a separate process for obtaining genetics information and variant calling. So basically, this data is integrated later. So we have different analyses and they are integrated later. As to quantifying how much the genetics will influence our results, that seems like a hard problem, and I don't have an immediate answer to that.
Okay, but just to be clear, so the starting point of your analysis were all cases which were all patients which already have this type of disease, right? Or
Yeah,
is it also
either,
healthy control?
Either healthy or controls, yes, or patients that have this disease, yes.
Okay, so you can already assume that the genetics part is essentially passed for the patients, because if they have the disease, they will probably already have the
Yes.
genetic component
Yes.
in a sense. Okay.
Yes.
Now this is a hard case because it's probably a continuum between people who are susceptible because they have a strong genetic component on one end, and on the other hand, cases where it's just the environment somehow causing most of the disease, but also, and there's a continuum in the middle, obviously. So trying to deal with this continuum, I think, would be an interesting expansion of what you have been doing so far.
And also time is very important, because I've read in the literature that the time evolution of the microbiome is basically it varies widely based on whether the patient is ill or healthy. And so in this work, we essentially only considered a snapshot of patients at a certain time. But actually, it seems that there is a very strong correlation between temporal variance of the microbiome composition and diseases. So that would be another kind of literally another dimension to extend our work to.
Thank you. Are there other questions? Actually, I have a very quick one. It's more a curiosity, since I'm not very familiar with the field, and it's partially related to what Professor Tosatto was saying at the very end, and it's, let's say, related to the data set, basically. So what I'd like, I mean, you spent some time on talking about it, but the question, the simple question would be, how is it a public data set? How did you collect it? How much is it a good, in your opinion, a good representation of a broader, larger domain, basically?
Yeah, thanks. So basically, the data sets are obtained from public repositories. There is this collection of databases. It's basically a metadatabase called the GM repo, the gut microbiome repo, which contains links and the metadata to various sources of microbial data. And the process of collecting this data was basically, I first filtered from this GM repo for patients that have the disease or healthy controls. I tried to obtain studies that seemed high quality and thoroughly researched. I also try to emphasize that there should be some variance or diversity in the metadata. So males and females, old and young people, people from different countries, etc. And then I collected a list of patients and studies for which the data was available for download. And then I needed to download the data and also needed to massage the data a bit because they are in different incompatible formats. But after that, it's only a matter of downloading the raw data and then feeding it into our metagenomic analysis software or another one. And basically, it means that we have raw DNA reads going in on one side, and on the other side of the software out comes a big table of relative abundances, and this is how the data is obtained.
Thank you.
Thank you.
So, time for a very last quick question.
Okay. From a biological point of view, do you think that your database, your software, the tools that you developed may be applied to a follow-up of these patients, because you may be able to identify biomarkers which change if the patient is subjected to a specific diet. And so if you identify biomarkers, these may be used for people that have mild symptoms as are at the beginning of the disease, if you can say something on this. And also for other diseases, fecal transplantation is used, like for Clostridium difficile infections. Do you think that this is a strategy to go for, and your tools may be applied to see how patients recover?
Thanks for the question. Yes, so as to biomarkers, yes, I think this tool generalizes quite well because it was written in a not with a specific kind of diseases in mind. We only use IBD as an example because we suspected that since it's a disease of the gut and the intestines, it will have a strong biological signal associated with it. But because it's a completely general tool, it can apply to any sort of diseases, and for identifying biomarkers, it's probably only a matter of collecting enough data and training the models accordingly on data that is sampled from different kind of patients. So yes, I think this could generalize quite well to identifying other biomarkers. And as to fecal transplantation, if I recall correctly, our company actually has plans for recommending such treatments. So we actually talked a little bit about with our medical and biologist teams, and it seems that yes, in some severe cases, it is necessary to recommend this kind of treatments. So yes, that is also one thing that we do.
Thank you. Very interesting.
Thank you.
So thanks. Congrats to both candidates on your work and presentation. So now I'll start a breakout room, and so you'll still be online. I mean, you two candidates. So please shut down your microphone. Even in the meantime, we discuss with the committee, and then we will be back live in I don't know, 10 minutes or something like that. Okay.
All right. Should we just stay here in the meantime?
Yeah. You stay in the. Both of you. You can stay on this. In this.
Okay. Thank you very much.
Okay.
Great.
So let's see if it works. Just 1 minute. Okay. Well, let's wait a few minutes. One of the two candidates is missing. Okay, we can. We have just the first candidate. We can start with the first one, or we have to wait both of them. Marco.
Oh,
Okay, so
if you want, you can start.
Okay, let's start with Claudia Fracca in the meantime, and then hopefully also the second candidate will show up. So, Miss Claudia Fracca, you have passed the final examination with the score of 110 out of 110 and honors. And with the authority vested in me by the Magnifico Rettore, I award you the degree of Master of Science in Data Science.
Thanks.
Congratulations.
Thanks.
So hopefully you can soon you can have also a better and more proper celebration. But anyway,
Thank you.
Congrats on your work.
Congratulations.
Thanks.
And Árpád is not here. What should we do? Let me see. Wait, okay. Let me try this way because he was. Okay, here we are. Okay, Árpád.
I'm back. Yes, thank you.
Good. We are all here now. Okay, just 1 second. Okay, Mr. Árpád Goretity.
Yes.
Hope that the pronunciation is right. You have passed the final examination with a score of 110 out of 110 and honors. And with the authority vested in me by the Magnifico Rettore, I award you the degree of Master of Science in Data Science. Congratulations.
Thank you very much.
And good luck on your next steps.
Thank you. And I would like to thank the entire university and the professors for a lot of help and flexibility. And best of luck and to you too. And I hope you will have a great summer.
Thank you.
Thank you and congratulations.
Congratulations.
Thank you.
Congratulations.
Congratulations to both of you.
Thanks.
Okay, now you deserve good holidays. Enjoy.
Thank you very much.
Okay, I can close the session then. Bye.
Goodbye.
Bye-bye.
Bye, everybody. Bye-bye. Thank you very much.